So we're going to be talking all about EMBL EBI data resources, and the aim of this webinar is that it can give you a sort of general understanding of what the EBI provides in terms of data, what you can access, and also training to go along with it to help you get into more depth in terms of different resources. Before we properly start, I actually just wanted to put a few poll questions out there to learn a bit about all of you and how you may or may not have used any resources before. So let's get started with a few polls. There's just 3 questions, all about whether you've used EBI data resources before, whether you have used whether you've submitted to data resources before, and whether or not you've had a look at our training. So these are the main things that I'm going to be talking about today. And I can see lots of people voting away to the various questions. So don't feel like you have to, but please do if you'd like. Okay, so a bit of a mix in terms of whether or not people have used data resources before. And I added the unsure answer there because I know certainly quite a few people have used some bioinformatics resources, but they're not quite sure whether they're EBI related or not. So we're going to talk a bit about that today. Okay, yeah, so a bit of a mix on that. For the submitted, whether or not you've submitted data to a resource, that's coming out largely no. So that's interesting. And we're going to talk lots about submission later on. And training, that as well is looking like lots of you haven't had a chance to access the training yet. So I'm going to show you all about that too, and allow you to know where to look for things really, and to show you what's out there. Okay, so right, thanks everybody for answering those questions. So as I just mentioned, those are the things we're really going to be focusing on today. We're going to be talking a bit about what EMBL EBI does, its role as a data provider. We're also going to be talking about how to find and how to access publicly available data through EMBL EBI data resources. And I'm going to show you a demo so you can learn a bit more about that. And as mentioned, I'm going to be talking about submitting to data resources. And I get lots of you may not be at that stage yet, but it's something to think about for the future. And then finally, onto the training aspect of what you can find and where you can get it, and how you can work through the training that we make available. Okay, so a little bit of an introduction to EMBL EBI. So we are EMBL's European Bioinformatics Institute. This is where I'm sat today on a not quite so sunny day in the UK. But EMBL actually has 6 sites right across Europe, focusing on things like bioinformatics, structural data, neurobiology, all sorts of things. But the EBI is focused solely on computational side of biology. In terms of our mission, I'm sure you'll understand by now, we make lots of different data resources available, so lots of different types of data, and I'm going to talk lots about that. We also have research teams. We do training. This is what you're going to hear about as well. And we're also get involved with industries, for example, pharmaceutical companies, and work with them in terms of bioinformatics across Europe, really. So today I'm going to be mostly focusing on the data resources. I may refer to them also as data services, meaning the same thing. So these services, I wanted to show this map. You can actually see it on the EBI site. But I really like this because it highlights that whilst we are the European Bioinformatics Institute, the data and the resources that we make available are used across the world, and everyone is welcome to do so. And I'll be talking a bit about open data as well during this session. So I mentioned that some people are sometimes unsure about whether or not a resource is an EMBL EBI data resource. And I have a few that you may have heard of before, and you may or may not know whether they are EBI resources. So one that a lot of people have used, particularly in their studies before, may well be Ensemble. Ensemble is a genome browser, and it's a great place to look if you're trying to learn about a specific gene and find out more information about it. Uniprot is another EBI resource, which is all about proteins, so protein sequences and function. This is the place to go if you're interested in learning more about a specific protein. So you can even just go straight ahead and put the name of a protein into the search bar, and you'll find out all about it. And the last one I wanted to highlight is one that I'm sure many of you have heard about over the last couple of years, AlphaFold. So for those of you who don't know about AlphaFold, it's all about predicting protein structures. And this is in collaboration with Google DeepMind. But these are just a few examples of data resources where you can go straight to them and access loads of different types of data. This is a slide I wanted to spend a bit of time on because this shows you all of the different data resources that are available. And you will see at the top of each of these different colors are different fields, different areas of biology. So we start on the left, all thinking about chemicals, drug discovery. So EMBL is the place to go if you want to know more about a chemical structure. Metabolites, all about metabolomics, and then open targets for drug discovery. So there's lots in terms of chemicals, different resources here. Then we have genes. So I talked a little briefly about Ensemble, the genome browser. There's also things like Magnify for metagenomic data. Then we have RNA data in RFAM and RNA Central. So lots of different types there too. Proteins, we have Uniprot, as I mentioned, which is a great first place if you're looking to find out more about a protein. But there's also places like PDBE to look for experimentally derived protein structures as opposed to AlphaFold for predicted structures. There's also PRIDE to look at mass spectrometry data. So all different ways of looking at proteins. Then a slightly newer area, we have the imaging section, and in particular the Bioimage archive, which has all different types of images from biological experiments for you to access. And we also have the genetic variation disease data. So for example, COVID-19 data platform, and in fact, the one that's missing from here, the pathogens portal. It's all about different pathogens, and it's a great place to start if you're looking for that kind of thing. Then we have model data, so mouse informatics, mouse model data, and then onto the literature and the knowledge management side of things. So for example, biomodels for mathematical modeling, the GWAS catalog for genome-wide association study data. So all sorts of things. And importantly, all of these data resources are completely open. So you can go to the resources website and search the data straight away, just jump straight in. And there's one exception to this. And this is the one that I've starred here, the European Genome Phenome Archive. Now, there's an exception for this one because it has what we call sensitive data. So in this case, it's patient data relating to patients in hospitals, for example. And because it's sensitive, related to individual people, instead of being open, there's information, there's metadata about the data that's available. But as a researcher, you're able to apply to access the data. So that's one exception. But other than that, all of these other resources, you can go straight to the site and have a look around. You'll also see this big arrow with various animals and wanted to highlight here that it's also not just about human. So we've obviously got a big human here, but there's also loads of other different types of species represented, as well as, in fact, various plant species too. So not all resources have a variety of species, but many do. And so that's something to keep in mind if you're not working with human data. So we have all of these different resources, but they don't work individually. They don't work in silos. You will see that actually lots of them interact with each other. So this diagram on the right here, around the edges, you will see each of these are names of different data resources, and the lines going between them show how they interact. So for example, if you search for a protein in UniProt, you may well end up clicking a link that takes you to a protein structure in PDBE, the Protein Data Bank in Europe, which may end up taking you somewhere else. So they link together, and this means that you don't necessarily need to know which resource to go to next. You don't have to go to it yourself to search for data. You'll be led straight to it. A really important question to think about is, where does the data in these resources actually come from? And usually it comes from researchers, so people like you who are working in labs, for example, doing experiments, generating data, and they submit to data resources, and the EBI data resources make it openly available. I'm going to talk a little bit about the research data lifecycle, which may be something that you've heard about before. But for what we want to talk about, I'm going to start at the plan here. And this is generally something that you as scientists will be doing. You'll be planning your experiments. You collect your data, you process it, and you probably analyze it. And at that point, you may keep it somewhere. For example, if you work in a university, there may be specific places for you to host that data. Alternatively, you can also submit it to a resource which will preserve it for you. Also, some data resources do some elements of analyzing as well, so it depends on where you submit it to. But they importantly preserve it, they keep it, they share it, as we talked about, sharing it openly, which then allows other scientists to access it and reuse it. I'm going to talk a bit more about submission later on and talk about reasons why you may want to submit, because we have quite a few that we think about in different ways. And I think that will hopefully give you some ideas about why you may want to submit your data. So to look first at the EBI website itself. So rather than going to specific data resources, I'm looking at going straight to the EMBL EBI homepage, and you will see down at the bottom here, it's simply ebi.ac.uk, and that will get you to this homepage. I'm going to show you a few slides now, and then I'm going to show the website in action in a moment. So you'll find on the homepage, there is highlighted in orange here, a big button that says Find Data Resources. So if you would like to find a specific resource, this is the place to go. And you will see also this Explore All Our Data Resources and Tools button. And this will show you a big long list of all the different resources and the different tools available, and a little sentence about all of them. You're also able to search at this point. But if you're completely new to EBI data resources, I really recommend having a look at this list and exploring it a little to see a bit more about the kind of data that's available. We also importantly have this big search box right at the top, and you'll see it says find a gene, protein, or chemical. And you can paste, for example, a gene name, a gene symbol straight into there, and you'll find loads of information. So in this example, I've searched for a gene, and you can see there's a huge amount of information available, and I'm going to show what this looks like on the website now. So if I navigate to the homepage, hopefully you're seeing the homepage too, and I'm sure Ajay will let me know if you're not. So as I just mentioned, there's this big Find Data Resources button. So we're going to take a look at that first. And I recommend this Explore button here as well. Now, at this point, there's a lot to look at. But if you have an idea about what you're looking for, then you can go ahead and just search. So for example, I know I'm interested in GWAS data. I can put that straight in, and it will send me straight away to the GWAS catalog up here. So I can click through and start exploring. So if I'm looking for something in particular, I can jump in there. I can put it into the search bar, or I can explore some examples that they've suggested to me already. So that's if you want to go directly to a specific resource, and you can take a look through this list to see what's there. However, for lots of you, I'm sure you are looking for information about a specific gene or a protein. So we're going to go ahead and pop the name of one into this search box at the top. So in this case, I put MSTN in here. I'm going to leave this as all, but if I wanted to, I could be more specific about what kind of information I was looking for. And then just hit search. And at that point, it starts loading quite a lot of data, okay? So the first thing I wanted to show you was this gene and protein summary. This is a really great place to start if you're trying to just get a bit of information about it, try and understand about the gene that you've searched for. Okay, so if I look at the gene summary page, you will see there's a bit about, well, its name, first of all, a bit about where it is, so chromosome 2, the location, some variation data, orthologs. You can link through and follow any of these. And there's loads of information there. But it's showing me right at the top here that this is coming from Ensemble, genome browser, and I can also link to that to see more information. There's also lots more on these different tabs. So a summary of the protein information is here. And again, you can see where this information is coming from. So Interpro or Uniprot. And there's lots you can work through if you're looking for a summary of information. But we'll step back and we'll go right back to the initial search results rather than the summary. And you will see down the left-hand side are all the different types of data that's been returned for this one gene that I searched for. So if I knew I wanted to look at structure, so protein structure, I can go straight for that. And there's a variety of different structures that it has shown me. And I can see that these are coming from AlphaFold, so predicted structures. And clicking on one of these will take me right through to that page within the AlphaFold database. So this is a great way, as I said, of if you are looking for something in particular, gene or protein in particular, it's a great way of getting a good overview of the kind of data that's available. And for a lot of genes, it's a really broad amount of data. Once you've been a bit more specific about what you're looking for, you can also be more specific about the type of organism that you're looking for as well. And you can use filters down the side, and then it will open up if you're looking for something that's not showing you straight away. So if you're new to these data resources, I would really recommend picking your favorite gene, plugging that straight into the search box right at the top of the EBI homepage and having an explore and seeing what kind of information, what kind of data is available to you. So just jumping back to my slides. One thing I wanted to mention before I move on is about programmatic access. You may also hear this as APIs. So this is the way that you access the resources programmatically. Most of the data resources have some kind of programmatic access available. This is obviously particularly useful if you are, for example, writing some code that would allow you to grab some data from one of the resources and bring it back and analyze it, rather than going to the browser, maybe downloading some data and analyzing it that way. So it's a great way of building the data retrieval into your workflow. So if this is something that you think you might find useful, all the resources will have something that says either programmatic access or API on one of their pages in the About section, and there'll be details on how to use it there. However, there's also lots of information about programmatic access within the training pages as well. I'm going to show you much more about the training in a moment, but you'll see there are things like recorded webinars on programmatic access for using specific resources, or some courses that you can work through. Quite a few of these have things like Jupyter Notebooks or Google Colabs in this case, that allow you to work through what happened in that training, so you can follow along with the code at the same time. And this is also a great way of, if you're just getting started with a programming language, in this case Python. It's a really nice way of seeing how the programming language can be used to access data. But as I say, we'll talk a bit more about training later on. The other thing that it's good to think about when you're using, well, really any publicly available data resource, is keeping track of what you're doing and citing the databases as well. So when I say keeping track, I mean it's a really good idea if you use any data or download any data to keep track of when you did it. So for example, you might want to record the date, but also data resources often have data releases. So there'll be a number that comes along with the data release. The reason this is important is that sometimes the data can change. So if more data is submitted, it may change what's already there. And past data releases are available, but it's not always what's visible on the browser. So it's really good just to keep track of things so that you know when you did it, how you can access it, whether you need to go back to an older data release to check what you've done before. So just like you would do in the lab, keep track of what you're doing as you're doing it. As I mentioned as well about citing, so just as you would cite a paper that you use information from, data resources can be cited too. And you will find information about this on each of the different resources. So this is a screenshot here from the Uniprot website, which just gives you a bit of information about the latest Uniprot paper and says, please do cite that if you use the data from the website. So that's just something to keep in mind for when you are using these data resources. Now, I said before I would talk a bit about submitting data. I know that some of you may not yet be in the position to submit data. Others may be producing data and want to think about this as well. But I wanted to talk about a few reasons why you may want to. So I've broken these down into some different areas. And the first one I wanted to think about was reasons that it's good for you to submit to a data resource. I don't know how many of you have been in the position, I think I certainly have, of having produced some data some years ago and then losing track of it, and you're not sure where it is or what's happening to it. The great thing about submitting data to a data resource is that it'll always be findable. It'll always be accessible to you to go to the site and say, yes, that's where my data is. I know it's safe, and I know how to find it, and I know how to share it with other people. And I think that's a really useful thing for a lot of people. It's also a really great way of sharing with other people so that you can get some recognition. You can say, yes, this is my paper that I've written, but also here's the data that went along with it, and you can look at it and you can explore it too. And that kind of leads on to that scientific aspect of reproducibility, traceability. Other people can really see what you're doing when you've published the paper that goes along perhaps with the data, and they can get the whole picture. And then one of the ones that's really important for me is that when people generate data, when they come up with the idea of an experiment, they have a question in mind and they're trying to answer that question. But other people may have different questions that they could ask the data that the original investigator may not have thought of. And so it's great to be able to put the data out there to say, right, I've done my exploration of it for my reasons. Other people can now take a look and see what they think and maybe ask their own questions to the data. So here again, just showing the screenshot of the EBI homepage. And next to that Find Data Resources button that we looked at before, you will also see a Submit Data button. And that will take you through to this data submission wizard. And what this does is it asks a few questions to you about the kind of data that you're interested in submitting to a resource. And then as you answer those questions, it guides you to the right resource and provides you with information about submitting to it. So if you think, yeah, that's what I want to do, I want to submit my data to a public data resource, I want it to be openly available, but I'm not sure which one, this is a place to go, and it will guide you to that, to the right place. I wanted to just come back to this life cycle again. Now we're thinking about submission. Whilst submission is around this point of preserving and sharing your data, it's actually something that really you need to start thinking about from the start, from planning your experiments. The reason this is really important is because along with data that's submitted, we also need metadata to be submitted. So metadata is essentially the data about the data, the information that makes the data understandable. So it could be details of the protocol you used. It could be things like the organism that you did the experiment in, that kind of thing. It's all about other people understanding what you did. Different data resources require different types of metadata because the data is different as well. So if you work out where you want to submit your data to at the point you're planning, you can make sure that you're collecting the metadata that you need to submit. There's lots of information about data submission on all the different data resources that you can submit to. But if you're not sure and you're at the stage and you're thinking, right, well, I'm planning what I want to do, but I'm really not clear on what I'm going to need in the future to submit my data, there are people called biocurators who work for data resources, who are the people behind the help desks of the resources, who are there to answer your question. So if you're at the planning stage and you have some questions about submission, about the kind of thing you need to collect, the kind of metadata you need to collect, you can message them, you can email them and say, this is what I'm planning on doing. Can you help me with my question? And they'll be very happy to do so, particularly if you're at the planning stage, because that gives both you and them the time to work out your question. Okay. So we talked a bit about different EBI resources, about submission, and the other main thing I wanted to look at today was all about training. So hopefully you've had a look at the EBI training website because you got to this webinar, which is part of the training program. But we have a lot of different training available. So I want to show you the different things that we have. So I'm going to show you a few slides first, and then we will take a look at the training website too. Okay, so on the EBI training homepage, you will see these 3 boxes right in the middle. And I'm going to be largely focusing on the on-demand side of things, but I also wanted to mention the live training as well. So in live training, we have webinars, live webinars, so just like the one you're currently at. Today's webinar is quite broad. It's a general introduction to EBI resources, but other webinars that we have tend to be more specific. So for example, how to use a specific data resource or an introduction to an area in bioinformatics. So for example, the next webinar that we have coming up is an introduction to the new look Ensemble genome browser. So if Ensemble is something you've used before, I'd recommend this one because you can learn all about the new look and feel and how to use the newly released Ensemble. So it's all very different, but looks great. So I think that's something that'd be good to learn about. But we also have lots of other different webinars available. So, and they are constantly being updated, new ones being released. So LinkedIn is probably the best way to keep track of what we have coming up, or returning and looking at the website occasionally. I'm actually going to start in the middle of this slide because we have those live webinars that I've just mentioned, but we also have recorded webinars. So everything that happens live in terms of webinars, they're all recorded and they go straight on the EBI training website. So you can watch the recording, you can access the slides, and you can work through them at your own pace. Also, if you need to go back and check something, the recordings are there for you to do that. At the moment, we have something like 200 webinar recordings available on a really broad range of topics. So I recommend having a look around and seeing what might be suitable for you. Then we have on the left here, online tutorials. Now these are self-paced short e-learning courses. So just like the webinars, they are a combination of introductions and how to get started on using different data resources. But there's also ones that are more introductions to different concepts in bioinformatics. So for example, we have here Bioinformatics for the Terrified. So if this is a topic that's completely new to you, I recommend starting here and you'll learn about bioinformatics from the start. But we also have other conceptual things like functional genomics, like all sorts of things, data management, which is probably a good one after the data lifecycle I was just showing. But then the other side of things are the data resource introduction. So, for example, there is a practical guide on how to use AlphaFold, which is very popular at the moment, or how to get started using biomodels, the mathematical biology data resource. So it's all there, and I'll show you in a moment how to find these things. Then we also have what we call curated collections. These are like really big online tutorials. They're combinations of videos and interactive elements and text and images that take a few hours to work through, but they introduce you to an area of the data-driven life sciences. So, for example, we have here finding and using publicly available data, but we also have one on chemical biology, an introduction to that topic. So there's loads of different collections. If you're not sure where to start, these are a great place to go. Lastly, we have course materials. Now, these are materials that are used at our week-long training courses that we host here at the EBI. So these are courses that people apply to, and there is a cost associated with them. But all the course materials that we use for those courses are made freely and openly available. So I'm just going to skip through these slides because I'm going to show you what it looks like on the website in a moment. But before we look at that, I wanted to show you about keeping track of your learning. So all the training is completely openly available. You can go straight to the website. You don't need to log in. You can just access it all straight away. Anyone can do that. However, you'll also see that there is an option to log in, to register and log in. You can choose to do that if you would like. If you do, there's a few things that it allows you to do. So for example, you will see there's these buttons where it says marked as favorite or mark as complete on pages of our tutorials. What this does is if you're logged in, it will keep track of your learning, and you will have your own personalized My Learning page where you can see what it is that you've been doing, and you can jump straight back in. This is also quite a nice way of providing evidence of your learning. So you will see that there is this completed tab, and on that tab will show any of the on-demand training courses that you have completed. So for example, if you wanted to show a supervisor what you had been doing, you could print this page and it will provide evidence of your learning. If you're logged in, you're also able to create playlists of training. You will see when you're logged in that it's possible to add to playlist on all the different types of on-demand training. And at that point, you're able to create your own playlists. It's possible as well to share these with anybody. So even if they aren't logged in, you can get a link for your playlist and share that link with them. So for example, if you are a lecturer or you're working in a study group, you could create a playlist of training and share with your students or your colleagues. And that's a great way of signposting other people to specific training. I'll show you in a moment how to find the playlist function. So at that point, I'm going to jump again from my slides to the EBI training website. So I think you've probably all been here before, as I say, to get to the webinar. You'll see on the front page, we have some featured courses, so a few different things that are upcoming. And then these boxes that I mentioned, in particular, the on-demand training will take you through to the different elements that I have been showing you. So you'll see we have lots of different things available. Just over 200 recorded webinars, 80 tutorials, and quite a few collections. So you will see that you're able to go straight through to any of these, and you can just jump in, and the watch video button will allow you to watch the recording. If it's a short e-learning course you're looking for, there's lots available here. And in a very similar way, you can use the blue button to enter the course. There's a bit of information on the front page of each one of these that explains what it's about, who it's for, what the aim is, what you hope to achieve by completing it. And then you can jump straight in and get going. If you're completely new to EBI resources, I recommend this one, A Journey Through Bioinformatics. And what it does is takes you through various different data resources and provides you with a challenge. So a challenge and a hint on how to solve it, and then a little quiz to check that you're getting the challenge correct. There's a bit of extra stuff here because I've already done this before, so it's telling me how I did. But there's lots of different challenges there for you to explore. And then the last thing on here I wanted to show was the collections. So I mentioned these are a really good place to start if you're looking for a specific area of biology. So for example, you might be a plant scientist. And you will see when you jump into this, there is loads of different training available related to plants, But the advantage of the collections is you don't have to know about which data resources you may want to know about. You just need to know, I want to know stuff about plants and data sciences for plants. And each section will take you through the relevant resources associated with that topic. So as I said, great place to start if you're not quite sure what kind of training you're interested in yet, other than a general area, general field. Now, while we're on this site, I just wanted to show you up the top here, we have the My Learning page that I mentioned that keeps track of your learning if you're logged in. And then we also have this My Playlists section here, which is where you can go to create playlists and share those with other people. Okay, so that is the training site. And I'm just going to jump back for a couple more slides and then we will have a look at some questions. So you may want to get some help while you're using the EBI resources. And I mentioned about biocurators, who are the people often behind the help desks. Now you can fill in the support and feedback form that you'll find on any EBI resource, or indeed the EBI main website, and you can pick from the list the resource that you're trying to ask your question about, and that will find its way to the right person. You'll also see on different resources different ways of doing this. So it could be a little envelope or a feedback button, but they also all have lots of information about how the resources work. So there's loads to help you with your problems there. So just before we move on to the questions, I wanted again to highlight the next webinar that's coming up, and that's the new Ensemble Genome Browser that's available to register. And you will find all our webinars at this link. So with that, I think we can end the presentation and see if we have any questions.
But there are a few open questions, and I think they are good to be shared. So the first question which I can take is, can we openly share this data online for educational purposes?
So you will see on the training site and on other EBI resources, you'll see here something that says CC, usually CC BY. And this is the license that the data and the training is made available under. And CC BY means that you can take the data or the training materials, do whatever you like with them, as long as you say where you got them from. So they're completely open to use. You just have to say, I got this from this data resource or from the EBI training site. But they're, other than that, completely open for you to use, particularly for educational purposes, but for research or whatever it is you're trying to do.
Thank you. There's one question about when someone submits the data, how the information is checked for validity.
Yeah, so this depends on the actual resource that you're submitting to. But essentially the biocurators run the data and the metadata through a series of checks. And actually some of the resources make these checks available publicly, so you can see what it is that you have to kind of match up to. And then depending on the kind of data, some of these are actually reviewed like by a person kind of going through them and checking them. It depends a lot on the kind of data, the resource, but they're the people who work through it either in an automated way or manually. It does depend, but yeah, this is why I think it's a great idea to get in contact with biocurators and to explore the help that the different resources give about submission, so you can really see those details. And it's something they do want to make really clear, because it makes everyone's lives easier if they can see exactly what the checks are and what you need to include with your data.
There were a few questions about if people can access EBI resources using programming languages.
Yeah, so let's have a look at the training site to start with. So if you put in programmatic access to the search on the training site, you will see a variety of different, usually recorded webinars, but a few tutorials that explain how to use programmatic access for different resources. So for example, I mentioned the Uniprot using Python, but there's a range of them here. But what you can also do is, alongside that, go to a resources website, and within the help, you will find information on, for example, here, programmatic access, and it lists all the information required to do that. And in a lot of cases provides quite detailed examples that you can edit to use yourself. So huge amount of information there. If programmatic access is completely new to you, I would recommend the Uniprot with Python webinar as a way of kind of understanding how they work in general and getting into the idea of it. But yeah, if you've got a specific resource in mind, there'll be some information and some training available for you here.
So one question is about, is there a database when we can input our small molecules and screen multiple transporter proteins?
So let's go to the EBI homepage and we'll look at the find resources. So small molecules. There we go. So Unichem is the place for you. There is also a tutorial that goes along with this that explains all about what Unichem is. So if you put Unichem into the EBI training search box, you will find a tutorial that takes you through that as well. But I think that's a good place for you to start. If you then go, we're back on the training site. If you pop Unichem, the great thing about the search in the training site is that it auto completes. So if you start typing something in, it will help you. So there's a quick tour that will get you going very quickly on how to use the resource. And there's also a recorded webinar that talks about how to use programmatic access for it as well.
Thank you. There were a few questions about getting a certificate after completing our on-demand tutorials.
Yeah.
About my learning, so if you could.
Yeah, so this, for those of you who log in to track your learning, you can go to your My Learning page, and it's not a certificate, but if you go to this completed tab, it will show everything that you finished when you did it, results of quizzes and things. So you can print this page and use that as evidence of what you have completed, so in place of a certificate, but it sort of shows you've been doing this via the EBI website and when you did it. I also just spotted one from someone who's saying what kind of scientific background enables people to work at the EBI. This is a massive range. So at the EBI, we have people who previously worked as lab scientists who then come in and they're the people who really understand the data that's being generated. So that's the case for a lot of people like biocurators. We have software developers, people who work in training. So for example, within our team, we have people who organize events. So they're not necessarily scientists, but they certainly have a place here. So all sorts of different jobs. Yeah, do check back in a couple of weeks and you'll see a bit more about that. One thing I wanted to show is that if the concept of biocuration is new to you, I would recommend having a look at this collection, which is all about the topic of biocuration. Talked a bit about the role of a biocurator that I mentioned, kind of work for the data resources. And this is often a way that that lab scientists, bench scientists kind of move into this more data field. because, yeah, as I say, they're the people who know about the data that's being generated in their respective areas of the life sciences.
Very good, thank you. And then there are some specific questions about training available in bioinformatic analysis, mainly sequencing, deep sequencing or ATAC sequencing.
I'm not sure off the top of my head.
I mean, we have a few courses for general sequencing analysis.
Yeah. Yeah, so this is actually particularly the case, a case where you may want to look at the course materials as well as the more self-paced or the recorded webinars, because there's a huge amount of material within these. These are, as I said, these are week-long courses that run, and all the materials get added to one of these sort of sets of course materials, and there's slides and things to work through, but there's also practicals that people have developed that you can work through yourself as well, and sort of instructions on how to do them. There's a question I just spotted on how can we access the access-controlled genome-phenome data.
Okay.
So if you go to the European Genome-Phenome Archive, the EGA, you will see there is a big button that says Access, and it describes how you can request access to data, and there's also ways of kind of searching to see what kind of data is there. So it breaks it down clearly for you to work through. So if that's something that you're interested in doing, I'd take a look at this access section of the EGA.
And someone is not sure about the training material that you mentioned, which are associated with live training. Is it possible
Yeah.
to see those materials?
Yeah. Let me just go back to the homepage here. So in live training, you will see there are the webinars, but there are also these courses, either virtual or at the EBI. So if you look through at one of these, you will see information about that course and a program that goes along with it. It's not much of a program on this one, but a program that goes along with it explaining what each section is. Then, once the live course happens, within a week or two after that happens, the materials will appear in the on-demand training section under course materials. So these are all courses that were usually 5 days long, that ran with people attending them. But at this point, after the course is run, we make the materials available. And you can look through those and use them however you wish. You may want to look through the slides or work through the practicals, or if you are teaching somebody else, you may want to use them for that purpose. So all the materials are there from those courses.
All right, thank you. And there are a few questions which came in. People want to learn programming. So I think we don't have a specific course just for teaching programming.
No, it's not something that we do, because there are more general websites that allow you to do that. But yeah, there's plenty, I think, that you can kind of use, and it'll introduce you to the basics of them. But I think if you're getting started with programming, the best advice I'd have, other than to do some of those general tutorials on different websites, is to come up with a problem. So that problem could be getting some data from a data resource and doing some basic analysis of it. And that's something you could use one of our training recorded webinars or something for. But by having that problem, it can really help you focus while you're learning, and also help you move along with that process. So I'd recommend one of those more general websites in conjunction with something specific that we have, and it will help you kind of progress and focus on how you're learning.
Great. So I think given the time, we can take probably one more question. And this is about data submission. There's someone is asking if they can submit their data without being part of a specific research team. at the uploading time?
Yes, yeah. It very much kind of depends again on the resource, but that's certainly the case, as long as you can provide the required metadata to go along with it. That's absolutely fine. Yeah. Actually, just one thing I forgot to mention about data submission. I mentioned about planning it ahead of time, but just because you have submitted to a data resource doesn't mean it's immediately openly available. There are ways of setting dates on it to make sure that it coincides with papers being released and that kind of thing. So there's ways to make sure that the data doesn't go out before you're happy with it sort of being made openly available.